Hi everyone, my name is Vincent Wu. I would like to first thank the selection committee, Dr. Nigel Fraser, and my advisor Mike for this prestigious honor. I am very excited to receive this award and share a little about my thesis work, which is focused on the HIV reservoir. For a quick background, HIV remains a major global health issue. As recent as 2020, there were 37 million people living with HIV and more than 1 million new infections. Since the introduction of antiretroviral therapy, or ART for short, people living with HIV can fully suppress viral activity, which results in undetectable viral load and immune restoration. However, such a regimen requires complete adherence, as viral rebound can occur if ART is stopped. This rebound occurs because of the HIV reservoir, which is this pool of affected cells that remain persistent even during treatment. And this persistence is in part due to the integration of viral DNA into the host cellular genome of long-lived memory CD4 T cells. And this reservoir is the main obstacle for a functional cure, which is defined as the prevention of viral replication without the need for ART. One of the functional cure strategies is known as shock and kill, where latent HIV cells are reactivated and then subsequently eliminated with either pharmacological or immunological methods. This strategy requires that we must first understand the HIV reservoir in order to successfully reactivate. However, there's actually no known signature that is unique to these infected cells. As such, increasing our understanding of this reservoir was the basis of my thesis work. My first main study looked at where HIV integrates its genome into the host cells in the context of different tissues across the body, as well as various cellular phenotypes, such as if the cell was tissue resident or if the cell is capable of recirculating in blood. I found that the characteristics of integration across all the factors were actually pretty similar. Instead, I curiously found that there were unique clonal populations based on if a cell was resident in a tissue or if it could circulate. This suggested the existence of sub-reservoirs. Knowing this meant that HIV reservoir studies must be done at higher single-cell resolution to not only parse through heterogeneous CD4 cell types, but also to inspect the infected cells in their specific immune/functional context. I then followed this up in my second main study by generating the first single-cell atlas of HIV-infected cells at their basal state in persons living with either chronic or ART-treated HIV. Here I used a novel methodology called viral ASAP-seq that could both identify as well as phenotype these infected cells with simultaneous epigenetic and proteomic profiles. Importantly, this was done at their basal and unmanipulated state and was the first demonstration of this capability. This revealed extensive heterogeneity in cellular phenotypes between and within individuals. I also found that some of the infected cells during treated infection displayed a poised signature for reactivation, while other cells actually had an increased threshold for reactivating. This suggests that any future functional cures need to take this into consideration in order to be truly effective in awakening and eliminating the entire HIV reservoir. Altogether, my results have painted a clearer picture of the HIV reservoir to help guide future curative efforts in reversing latency across the phenotypic diversity of the HIV reservoir. Once again, thank you again to the committee, Dr. Nigel Fraser, and Mike for this honor, and thank you all for watching as well.